Up to now, many of you may not have been aware of the fact that you can actually use light to kill cancer cells. Now, there's some serious limitation to what this treatment is called. Actually, it's called photodynamic therapy. There's some serious limitations to it. However, basically, in short, what it is, you sensitize the cancer cells to light. You shine light on the cancer cells. The cancer cells generate what's called ROS, or reactive oxygen species, and the cancer cells go bye-bye, in theory. However, it's had some serious limitations, which we're going to catch up on. But in any case, the Republic of Korea may have overcome those limitations. If their research translates into human trials to be as effective as they hypothesize, then it is an incredibly huge breakthrough in cancer treatment. Let's go right into the research as follows. Groundbreaking study sheds light on treated cancer. That's their title. The new study by researchers demonstrates a more holistic, light-based treatment to new cancer cells instead of surgery. Holistic was their words, not mine. In the first step, now keep in mind, we're going to look at a little bit of the definition of this just to get you up to speed. This definition comes from the National Cancer Institute, but it'll give you an idea of how incredibly promising this treatment is in the way it keeps healthy cells intact and only really targets the cancer cells. You'll see why in a second. In the first step of PDT therapy, otherwise known as photodynamic therapy, for cancer treatment, a photosensitizing agent is injected into the bloodstream. The agent is absorbed by cells all over the body. Now you begin to see the potential. Where can you shine light? Anywhere. But stays in cancer cells longer than it does in normal cells because there's a huge window of opportunity. Approximately 24 to 72 hours after injection, when most of the agent has left the normal cells but remains in cancer cells, tumors are exposed to light. The photosensitizer in the tumor absorbs the light and produces an active form of oxygen that destroys nearby cancer cells. The problem up to now, again, just reading straight from the National Cancer Institute because their explanation is really good. The light needed to activate most photosensitizers cannot pass through more than about 1/3 of an inch of tissue. For this reason, PDT is usually used to treat tumors on just or under the skin or lining of internal organs, blah, blah, blah. PDT is also less effective in treating large tumors because the light cannot pass far into these tumors. PDT is for local treatment and generally cannot be used to treat cancer that has spread and metastasized. Until possibly now. And they're using a new compound called Iridium, as well as matching it to the proper infrared wavelength, which is not used in different colors like blue or green. Infrared and Iridium seem to be an incredible, Iridium-3 I should say, complex. Let's go right into the study so you'll understand more in a second. This is just fascinating. Study parameters are as follows. Endoplasmic reticulum localized Iridium-3 complexes as efficient photodynamic therapy agents via protein modifications. DOI is as follows. Funding. This is the interesting part about it. Now, UNSIT is the Ulsan National Institute of Science and Technology from the Republic of Korea. It's funded by the Alzheimer's Disease Research Fund. Study participants, obviously, was in vitro. The cytotoxic studies use ovarian cancer and breast cancer cell lines as follows. Study length, not applicable here. Dosage and materials. They're incredibly well listed. Everything they have as far as in regard to being forthright in what was done in the study is upfront. Anybody should be able to replicate the exact same study that these guys did, or should say get similar results to what these guys had done. Results. And I'll also keep that PDF on one of my websites so you can pull it up yourself. It's basically studying the methods and dosages and all that other fun stuff. Results as follows. The results show that the red light absorbing photosensitizing, otherwise in this case infrared, with longer wavelengths significantly accelerated ROS production. Remember we said ROS is reactive oxygen species compared to blue and green lights of shorter wavelengths. Now this is their word, and I end it here because you really can't add any more to it than what they already did. This is in their words, from the researchers' words. Using infrared light that penetrate deep into the human body, we are now capable of killing deep cancer tumors without damaging healthy tissues. Voila, photodynamic therapy just got a major upgrade. Again, as well as keeping healthy cells intact. Now, where you can and can't shine light into the body, well, that'll be up to the researcher to determine. However, though, if this research does translate effectively into human trials, and I really, really hope that somehow we do some research in the United States in this promising cancer treatment, it can be used in a myriad of different treatments for different types of cancer and so on and so forth. Fast, effective, and extremely, extremely lethal to the cancer while keeping healthy cells intact. Again, Ralph Turchiano signing off. Photodynamic therapy, something if you want to talk to your oncologist with, or at least present them with the current research. Be kind of interesting to see what the results are, or I should say the response, I should say, that you get back. And I always hope that you find this information of use. Thank you very much for listening once again. I'll catch you all again in 7 days. Thank you.